Hello, my name is Jordan Schlautman, and this summer I researched the effect of leucine zipper capping on the vesicle assembly of fusion proteins. I was born and raised in Nebraska and am an upcoming senior at the University of Nebraska-Lincoln. There I study chemical engineering. This summer I flew down to the University of Florida to join the Chemical Engineering RU and to do research. This summer I worked in the Jiang lab under Dr. Jiang and my PhD mentor Juyon Shin. So why are we interested in vesicles? Vesicles can be used to create drug delivery systems which allow for the targeted delivery of drugs to specific areas of the body, controlling how long it takes for the drugs to be released and where. Vesicles can also be used to study artificial cells, which can be used for metabolism, transport, and chemical reaction chambers. These artificial cells can also be used to test new technologies. These uses depend on size, permeability, and other physical properties of the vesicles. Globular protein vesicles are currently made of two subunits. This consists of the globular protein M-cherry and a ZE leucine zipper, which is glutamic acid rich, and then a ZR ELP, where the ZR is an arginine rich leucine zipper and the ELP is an elastin-like polypeptide, which changes hydrophobicity with temperature. And combining these two, we can form a homodimer and a heterodimer. And these have a hydrophilic head, which like water, and end up on the outside or the very inside of the vesicle, and a hydrophobic tail, which don't like water, and end up in the inside. And these soluble proteins can form coacervates and then vesicles with temperature changes. And here is what the vesicle structure actually looks like. So what we are changing. The focus of my project was to isolate the ZE protein and to use it to cap the ZR-ELP protein. This would change the morphology of the membrane. We could then change the ratios of the amount of each protein we have and see how it affects the vesicle structure and function. This was done by using electron microscope, dialysis testing, size testing, etc. And so by changing the interactions between the subunits, we are then changing the packing efficiency. So as we can see, the ZE-ZR-ELP is a more cylindrical, narrow shape, and this is changing the critical packing parameter. And using this chart, we can see that the morphology is more of a vesicle, and the packing parameter is a 1/2 to 1, which is more thermodynamically favorable. And so we are looking at how the stability changes and the sizes of the vesicle changes. So making the proteins. So the E. coli, the plasmid DNA was taken, protein instructions were put into the DNA, put back into the E. coli. This is when we received the E. coli. And then we took the E. coli, put it into a growth medium where it multiplied, and then it ate the growth medium, made proteins, and then we harvested the proteins, which we can then use to make our vesicles. These are some examples of vesicles. Note the red ring on the outside and the dark red interior. And so those M-cherry proteins are fluorescing under the electron microscope here. And so to test the stability of these vesicles, we use dialysis testing. So inside the vesicle was about 0.3 molar of salt, and on the outside we did 0.137 molar of salt, which are physiological conditions, so conditions inside the body. And so this created a difference in salt concentration, and this created a desire for the water to go inside of the vesicles to have the inside of the vesicle salt concentration equal the outside. And so this could break the vesicle, leading it to explode out or to grow in size, which would then tear the membrane and make it into a coacervate. And so using this, the stability test looked to see if the vesicles were strong enough to withstand the outside forces and to remain vesicles and not turn into coacervates. And we did these tests for different amounts of ZE capping. I would like to thank Dr. Jiang and Juyon Shin for mentoring me this summer, and for the University of Florida for allowing me to partake in this RU program. I would also like to acknowledge the NSF funding that allowed me to do my research. Thank you.